Today you'll be learning about the process found in all cells called protein synthesis. Protein synthesis is important because it is responsible for your physical traits. Now, before you learn about protein synthesis, you need to know some basics about DNA and RNA, which are the main engines to the workings of protein synthesis. Deoxyribose nucleic acid, or DNA, is found in the nucleus. It is the carrier of genetic information that is used for growth, repair, and reproduction. Because the information in DNA is so precious and must be protected, it is unable to leave the nucleus, so it has to be copied and transferred. DNA is made of molecules called nucleotides. Each nucleotide has a phosphate bond, a sugar backbone, and a nitrogen base. The phosphate bond connects the nucleotide to others, creating the double helix structure that you see. There are 4 different types of nitrogen bases: adenine, thymine, guanine, and cytosine. Each base can only connect to one of the other bases. To prevent mistakes, there are 2 bases with 2 connection points, adenine and thymine, and 2 bases with 3 connection points, guanine and cytosine. The role of DNA is long-term storage of genetic information needed for development and function of living things. This means that it holds information which can be used to make proteins. RNA is like DNA in the sense that it has a sugar backbone, phosphate bonds, and nitrogen bases. But it only has a single strand and is instead used to transport information and create organelles within the cell. Another difference in RNA is that thymine is replaced with uracil. There are 3 different types of RNA: mRNA, rRNA, and tRNA. mRNA, also known as messenger RNA, is responsible for transporting information from DNA out of the nucleus and into the cytoplasm, where it is used for a multitude of processes. It is created in the nucleus, transported to the ribosome, and then degrades once it has served its purpose. rRNA, or ribosomal RNA, is a type of RNA that has been folded in a complex structure that helps to form the ribosome. It acts as a docking bay for mRNA and tRNA, allowing for messages to be read with ease. It can be known as the factory of the cell. It takes in information, imports the required parts, and creates the product. Ribosomes are found in the cytoplasm. It can either be detached and free within the cytoplasm or can be attached to the endoplasmic reticulum, creating the rough endoplasmic reticulum. mRNA is usually read by the detached ribosomes. tRNA, or transfer RNA, is responsible for carrying amino acids to the ribosome from the cytoplasm. It does not deliver the amino acid unless the correct mRNA code is matched. tRNA is found in the cytoplasm. It moves from the amino acid factory to the ribosome and repeats this process. You may like to pause the video now to write down these notes. The main things to remember are that DNA stores all genetic information for living things. DNA is found in the nucleus and it is unable to leave. Messenger RNA carries information from the nucleus to the ribosome. Ribosomal RNA is like a docking bay that provides connection between mRNA and tRNA. Transfer RNA transports the amino acids to the ribosome. Protein synthesis means to make proteins. This is the process of transcribing DNA into mRNA, then translating mRNA into amino acid sequences. Eventually, these will become proteins. Protein synthesis has 2 major steps: transcription and translation. Because transcription and translation sound so alike, if you remember that C comes before L in the alphabet, you can remember that transcription comes before translation. Transcription can be simplified to be known as making a copy or rewriting information. This is because in this stage, DNA is copied onto mRNA. Transcription can be simplified into 3 major stages: unzipping, copying, and transporting. All of these events take place in the nucleus. The first phase, unzipping, involves an enzyme called helicase, which unfastens the DNA, creating what is called a replication fork. It's given its name because of the appearance that the double-stranded DNA now has. The second phase of transcription is copying. This includes an mRNA strand copying the information from DNA onto its own backbone. Once copying has occurred, the third phase of transcription begins, which is the transportation. This involves the mRNA leaving the nucleus and entering the cytoplasm where translation takes place. The mRNA now journeys to the nearest ribosome. Translation can be remembered as making something legible or simplifying information. This process turns the mRNA strand into the amino acid chains, which will become proteins. Translation involves 5 different steps: connection, decoding, releasing, sliding, repetition, and stop. All parts of translation occur in the cytoplasm. Connection follows once the mRNA has left the nucleus and journeys to the ribosome, where it slides in between the ribosome's large and small subunits. Phase 2 involves another type of RNA called tRNA, which stands for transfer RNA. The tRNA helped to decode the mRNA brought to the ribosome. The ribosome lines up the first codon, which is a triplet of nitrogen bases, where a tRNA with the opposing bases, known as anticodons, match the mRNA codons. So it is referred to as the start codon. Phase 3 entitles the tRNA releasing its amino acid and starting the chain of growth. All of the amino acids are joined by peptide bonds. Phase 4, slide, is when the ribosome slides across to the next codon. Phase 5 is repetition of steps 2 to 4. Finally, phase 6 occurs. This is when a stop codon is read and the amino acid that detaches the chain from the ribosome is added. Stop codons can be one of 3 combinations: UGA, UAG, or UAA. What we've learned is that there are 8 steps in protein synthesis: unzipping, copying, transporting, connecting, decoding, releasing, sliding, repeating, and stopping. The final outcome of protein synthesis is the detachment of the amino acid chain and then twisting of the chain to form the protein. On very rare occasions, the protein that is made is incorrect. If the DNA strand is read incorrectly at the beginning, a nitrogen base could be wrong or missing. This means the mRNA is read wrong by the ribosome, which provides the wrong amino acid, then causing the wrong protein to be made. This simple mistake can lead to illnesses or diseases like retinoblastoma. This is when the subject's pupil turns white and involuntarily moves. A great way to remember difficult processes is to model them. So if you go to this link, there's an amazing tutorial on how to make DNA. And with that DNA, you'll be able to model the processes of translation and transcription.